In this video, we will discuss about different chimeric antigen receptors approaches. First, we will discuss uh, what are the different kind of CARs that are used and at what stage they are in terms of their clinical trials. So first, we will discuss about this, and so we will discuss about the NK cells, different source of the NK cells. First one is from the cord blood. It is readily available, and it requires these ex vivo expansions. And right now they are at the clinical stage, and they are being tested on the human beings who are affected with cancers. And second one is this induced pluripotent stem cells. They are highly proliferative, and but they have immature phenotypes and requires genetic manipulations, some kind of such as this. It will trigger CD16 mediated ADCC, that is antibody dependence cells cytotoxicity, and it is also at the clinical trials. The third one is this peripheral blood. It has this mature phenotype and it is highly functional as cytotoxic, but it also requires ex vivo expansions. And the another disadvantage is the donors are not readily available, and it is at this clinical trial, but it's not at the stage where it can be used for commercially on the patients who are affected with cancers. Another one is NK92 cells based therapies. They are highly proliferative and easy to manipulate and engineer. However, it reduced it has the reduced sensitivity to cryopreservations. And another disadvantage is it requires irradiations before it can be infused to the patients because of this NK cell lymphobia origin. And once it is irradiated, it has less persistence, less ability to survive after irradiations, and it has low ADCC due to the absence or low amount of CD16 expressions. And it is also at this clinical trial stage, but it is making a transition towards this clinical efficacy, that is it can for use of the patients commercially. The last category mentioned here is cord blood. This is also based on the stem cells, that is HSPC. It is readily available and possesses NK cells precursor. It has the potential to differentiate towards NK cells. It also requires ex vivo expansions and has very low CD34 positives. And it is at this clinical stage for non-engineered CAR cells. It is the first stage of the immunotherapy program, and one of the reasons could be because of this low ADCC and the absence of CD16, or the low presence of the CD16 in this kind of cells. And how these CAR cells are usually engineered. First one, we will discuss about the general CAR NK cells. So here the CAR construct against the specific tumor antigens is being transduced. It can be seen here, and it can target the tumors that has those antigens present, and it is not restricted to this HLA. It is MSC, the antigens information that can be detected by the MSC. So it is not restricted to that. But another disadvantage is that it requires complex manufacturing and no intercellular targeting. It cannot target intercellular regions. And whereas these T cells, receptors, NK cells, in this case the advantage is that it can target intercellular antigens, but main disadvantage is the HLA restricting killing. It requires the antigen informations that is compatible with MSC class 1, so that it can generate antibodies against those antigens. It also requires complex manufacturing and is not yet clinically tested. Next one is this bispecific and trispecific engagers. In this case, the NK cells are transduced with 2 or more tumor antigens that can be seen in this figure, such as CD16A and other tumor antigens, so that these NK cells efficiencies can be improved and it can target different tumors that has this different kind of antigens presence. But the disadvantage is that it has limited in vivo persistence. So these cells are not able to survive in vivo because of the less half-life time. It get easily cleared off by the human or patient's immune system. So these are some of the approaches, and what are the present stages of the different therapies that is based on NK cell source, different NK cell source. So if you need to understand more about their backgrounds, or need to understand more about their current status in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.